something a little bit different. Leo had recommended this package, PCAtools, and I took a bit of a look at it, but I guess today would be a session for us to sort of look through it together. And just, it has some functions for plotting and then some other things. And yeah, we'll just explore this package a bit. So I guess to get started, I guess we should do maybe a bit of background on PCA. So just PCA, of course, is like one of the more common dimensionality reduction techniques that we use. So maybe I'll annotate for this actually. So normally we'll have something like we'll have samples, we'll just call it S for now. We have a matrix of samples, and then we might have a matrix of, I'll call it G, so I'll just write it out, genes. You'll have basically as rows, you'll have observations. So in our case, it's often samples. And as columns, you'll have variables. And often we're going to be, obviously, we study genes a lot. So, and this is sort of inverted from, or it's sort of transposed from like a summarized experiment object that we're used to working with, where genes are the rows. But in any case, PCA, often the way we use it is we'll use it like, I didn't give myself very much room here, but we do it like this, where we have samples as it reduces it down to what we normally use. It's called a scores matrix, the PCA scores, which are different than the principal components themselves. I'll talk about that in a bit, but the idea is that we have samples still as rows, and then we'll have basically these PC scores, and we'll have a lot less of the principal components than genes. So, and each PC is explaining, the first PC explains the most variance, and then they're all independent, and they keep explaining more and more variance as you go down, cumulatively at least. So I mean, that's like a very basic intro, but yeah, in terms of the actual package, we can get started. So this is a bioconductor package. So I've already installed it actually on JHPCE, but yeah, you can always get the development version from GitHub as well. But I just installed the bioconductor version. I'm going to go through this example. So it's a differential expression example with some smooth muscle data. I'll be going sort of quickly through the actual biology, since I'm going to be focusing more on the package itself. So most of the time, I'm going to actually just sort of copy and paste this code. So we're just loading data from this airway package. I'm going to open up I'm using the VS Code app on JHPCE for this right now. So I'm going to actually load conduct and do it through here. Of course, we could do this locally as well, but I just tend to prefer working on the cluster. So I'm going to open up R and sort of just copy and paste some of this. Apparently, I don't have the package, so I should have checked that probably. Although it doesn't list it as a dependency here. I think it's just
Arrow is a bioconductor package.
Oh, it is, okay. Hopefully this will be quick, but
you need to use quotes.
Yeah. Get that installed over here. Actually, I'll probably copy this into a new script. So just call this developer. Copy some of this code, just getting the data loaded.
Nick, while it loads, can you go to your browser, then go back once on the browser?
Back.
Like to the bioconductor page for PCAtools.
Okay, I see.
Then if you scroll down to where the vignette sections, the documentation is there. You're right now looking at the HTML version of the vignette, but you can also open the R script.
Oh, that's true. Yeah, that's a good idea.
That way you can just copy everything.
Yeah. Okay. Good idea.
Instead of having to copy it like a bunch of times.
Yeah. So we've loaded the data. I believe the next section is just converting some of the genes to ensemble IDs, or symbols, from ensemble IDs. Just making sure I'm explaining the context here. So I do a bit of normalization as well.
I would recommend just copying everything. That way you just have it all and just execute some of the lines you want.
Yeah, that's probably faster. Yeah, so should have this package. Warning, warnings. Okay. So yeah, I mean, this package provides, I know there's a bunch of ways to do PCA already. There's like PR comp, and there's run PCA from, I think, Skater or Scran, forget. And then there's other methods as well. So this provides their own PCA function here that I'll run. Did I not load? Maybe I didn't load the, oh, of course, I didn't load PCAtools itself. I'll explain it. So you see one of the default, actually this isn't a default, but they have this remove bar variable here. So I guess it's already removing 10% of the variables. So it's calculating the variance of each, and then it's sort of just removing ones that are contributing less. So that should be configurable. I haven't checked it out too much, but there's, yes, a remove bar here. You can specify, I think null is probably not removing anything, which is the default for most PCA packages or PCA functions. And there's also options for centering and scaling. So centering is very standard. It's not going to really give you meaningful results if you don't subtract the mean for each of your variables, for each of your genes, I guess. Scaling, I guess, is a little bit different. So scaling is sort of putting each, if we're working with genes, it would be putting each gene on the same scale, so dividing by the standard deviation after you subtract the mean. So there's different reasons why to do this versus not do this. But if different genes are contributing differently and you want to sort of preserve that signal in the data, then you might actually want to not scale. So that's probably why they have the default as false. On the other hand, if you have a data set where you want each with variables on much different scales, and you want each to contribute sort of equally, you might want to set this to true. So, and then they have the rank parameter for number of PCs to retain. Obviously, like you could go all the way up to the full dimensionality of the data set, but no one does that because the purpose is to reduce the size of the data. Or that's one purpose. So actually, I wonder what the default is for this. I missed it. Rank equals null. So I guess, yeah, maybe it just gives you all the components. So I haven't actually explored these objects. It's probably not super useful here. But I guess this brings us to, after you actually compute the PCA, or you actually do the PCA, this sort of brings us to our first type of plot here, called a scree plot. So I actually don't have, actually does this work? I don't think it does. I don't have the X11 set up. I probably should have done that too, but we'll wrap this in a little statement here so we can look at it. And so we get this type of plot that shows a couple of different things at the same time. So obviously when you're doing PCA, each principal component is going to explain a different amount of variance of the total data set. With PC1, of course, always explaining the most variance, and then each subsequent PC explaining less and less. So you have two different pieces of data here. You have the variance explained itself in the bar plots, and then you have this red line, which is showing cumulative variance explained. So this is going to basically plateau at eventually 100%. And we could see that the first like 6 PCs are already explaining almost all the variance. So we're doing a pretty good job of summarizing the data set with just 6 PCs here. So, I mean, we can talk later a bit about, there's obviously methods for deciding how many PCs to retain, finding the elbow point and stuff like that, which this package does actually provide some functions for that. So before I get to that though, I want to go into some of the other plot types. So I guess the scree plot is the first one, and we keep going. There's what's called a biplot. So let me just show you what that looks like for the settings that they use.
Nick, you don't need to be using the PDF. Just use dev off after making plots. That will like make our plots.pdf actually complete.
I'm sorry, can you explain that again?
Just execute lines 79 to 81, skip 78.
Okay.
So like those lines like made the plot, but like the graphical device hasn't been closed. So you need to run line 81. And then at that point, I think you can open your plots.pdf. So it has here too, because like we didn't run them off after the first time.
Okay, cool. Yeah, I can do that then.
Mhm.
So these plots are kind of new to me, this biplot. So the idea behind a biplot in general is it's representing two different types of data here. In this case, we have our observations, like these are the points shown here. These are what are called PC scores, which I was talking about in the very beginning, which indicate basically if the observations are actually, maybe I have a little visual here or on the other. I found this, it's sort of a random, I found this from Google Images, but it'll be helpful. And just give like a quick shout out. This is from a lecture from Sujin Yang or Jing, but I found this sort of visual here. Actually, I guess it's file. But to understand those points on the previous graph, these observations, let's think of it like, let me annotate here. Let's have this is a very simple example where we have, let's say the x-axis is gene. We have one gene on the x-axis that's already been centered, by the way. So the mean is at 0 of its expression. And then this is expression of another gene 2. And then we have each point is going to be where each of our samples lies. So in PCA, the first principal component is this red line here. Let's say this is PC1. So the reason for that is because that's the direction along which most of the variance occurs for the observations. And then this is only a 2-dimensional data set, so there can only be up to 2 PCs. The second PC will be this. And by the way, I'm drawing it sort of right and up. The directions of PCs are not important. They're sort of arbitrary. So I could decide to say PC2 is pointing downward and PC1 is pointing leftward. It doesn't really matter. The meaning of it is not important. But so those are our actual principal components. Mathematically, it's the eigenvectors of the covariance matrix of the original data. But in any case, this is not what we're talking about when we're talking about scores, which are where these observations lay. The scores are going to be, let's say we're talking about, I'm going to try to circle this point right here. The scores are if you basically project each point onto each principal component. So you see that this point, if you project it onto PC1, it's positive. Essentially, it's pointing in the same direction as PC1 relative to the origin or the center of the data. So that would be the score for PC1. And same for PC2, you can project it this way. And it's also positive for PC2 in this example. So that's what the scores are when you're talking about PCA, and that's typically what people plot for observations. So that's just to give you some context on these observations. So that's one piece of data. The other piece is that we actually have genes here. So what do these vectors represent? So the terminology for that is they're called loadings, the PCA loadings. So that represents how much each gene is contributing to each principal component. So I think this is where I'm a little bit less sure, but I believe in this example, the loading for PC1 for gene 1 would be positive because they're pointing, like gene 1 is pointing, the positive direction is this way, right? And PC1 is, so I believe I have that right. This is where I'm a little bit unsure, but that's sort of the concept of a loading where you can see how much each gene is contributing to each principal component. So that explains sort of like the directionality of these. So like the CL or F115 is contributing more to PC2 than it is to PC1. And another sort of interesting way you can interpret these plots is that like you can sort of combine interpretations from the observations and the genes. So if I believe this is right, so this CL or F115 gene is sort of pointing in the same direction of this SRR ending with 17 sample is. So I think that should mean that C1 gene is higher than average in that sample. So you can sort of like jointly interpret both the observations and the genes in this type of biplot. So there's a lot to take out of this. Obviously, you can use it as like a pure dimensionality reduction where you just see where the observations are located and just to separate the data points. But there's more to these graphs that are more to interpret here. So yeah, I'm just going to go back to this vignette just to make sure there's certain things I wanted to feel more comfortable with presenting than others. So we've got the scree plot, we got the biplot. Yeah, I guess also you only get these genes if you do the show loadings equals true. That was what I was talking about. The gene loadings show which direction these genes go. So you can also customize, like I think they did 5 for each PC, or I think there should be a total of 10. Yeah, that's 10. So that's all customizable. And then some actual like aesthetic configuration as well. So let me see if there's anything else that's interesting here. I guess I can't rescale it. Yeah, because I haven't looked thoroughly at customizing this. But yeah, number of top loadings is 5. I think that's per component, since we used the default and we ended up with 10. So let's see if there's anything else. Very customizable. Lots of parameters to potentially edit here. It looks like you may even be able to draw. I'm actually never mind. I'm not sure what that ellipse argument is. Yeah, this is quite a lot of things to customize. And I was sort of curious if this is something I wanted to just check real quick, actually, if you can sort of assign this to, like, I don't know, this plot equals. I didn't check if these were like ggplot objects, for example, because that would be, hey, they are, okay. So that's really useful. You can, so in theory, you can do your own customization on top of this if you really wanted to, like add geomes, add, labels, and so forth. So very, very useful there. So yeah, let me remove this annotation. It's pretty distracting. So we got the screen plot, we got the biplot, and these pair plots. Yeah, actually, I didn't check these out yet. This is the one I was looking for. So there's also, we should be able to actually run this plot loadings with the data we already have. Should be, yeah, with the object we already have, I believe. So, oh, I did the dev off already, so I kind of messed that up, but.
No. Do it again. Do dev off after this.
Okay, and it should come up as the third.
It will just, you're remaking it. So basically, like PDF, open parentheses, quotes, rplus.pdf is run like automatically. You just need to run dev off afterwards.
Yeah, so this is sort of related to the concept I was just talking about with these loadings, how much each gene is contributing to each principal component. So it's taking the top, I forget how many we specified, but yeah, let me check that actually. Plot loadings, oh, it's lowercase. That's, I don't see it.
It was the second argument.
There's a second, okay, I just skipped right over that.
Let's get components, PCA sec length 5. So it's like how many components you want.
Okay, I just like, yeah. I guess I was like thinking that you could customize how many genes show up here in the loadings. Yeah, so generally, like, I think the magnitude of these plots is more important than anything else. Like genes with higher magnitude contribute more. Again, the sort of the directionality of this doesn't really matter. With positive versus negative isn't super meaningful, but this is one way that you can see like which genes are sort of driving variance. And of course, if you're looking at the first few principal components, if the genes are in the same direction as them, then they're probably driving a lot of the variance overall in the data set.
There's a third parameter to the function called range retain.
Okay.
The default there is 0.05. So anything below is like the cutoff for saying like how much each of these loadings, how much each of these features, so genes in this case, explain or contribute, I guess, to that principal component.
Okay.
Mm-hmm. So like that's why like your color range only goes from like basically minus 0.05 to 0.05.
Yeah.
But like I guess some of these principal components just have a lot of genes at that level, that range retain level.
Yeah. So actually,
do we have like, I guess, a use case for these like groupings, like Go or like do a Go analysis, or is it all kind of abstract?
in terms of like top gene.
Yeah, I guess like, is there anything that we can pull out like that's like biologically relevant, or is it just to better understand the PCs? Like,
Yeah, I'm not sure actually about that because this would be mostly talking about like variation, like just the amount of variance, I guess, important genes, not necessarily like what their function is or
okay.
why they're important. But I mean, I guess there is another type of plot, I guess, that maybe is more with like more straightforward interpretations, I guess. Maybe this eigencore plot where you can actually relate back the PC scores with metadata variables that you have. So like we could relate this to like, I don't know, diagnosis, sex, age.
Okay. Yeah, like this looks pretty relevant to, I guess, how we've interpreted these before, which is like, is PC1 driven by like a technical variable, like batch or something?
Exactly. Yeah. So yeah, that'd be the sort of equivalent in this package. Yeah. Other plots, I would have to think about how we would. For now, like to me, they're also a bit abstract. For now, I would have to think about how we want to use those. But yeah, I think also they also talk about like just internally these objects have different components. So actually, I have to remind myself because I thought the rotation matrix was the principal components themselves, but it's not. That would be the loadings.
So Nick, you have some questions in the chat.
Oh, yeah. Okay, let me check that. All right, so a couple things. So Cynthia is asking, plot presenting two sources of variation in the same space. Are the PCs of the genes independent of the PCs of the samples?
Oh, sorry, Nick, that is related with the previous upload that you were explaining. That one, no.
This one right here, right?
Yeah, that one. It's because it's not fully clear to me if I'm seeing two sources of variation in the same PC space or if these are independent. How to really give an accurate interpretation to these plots. I'm a bit confused with this one.
Yeah, this is a bit tricky. I guess I sort of like
You're talking about the genes?
Both. Yeah, I'm indeed to both because I'm looking here samples, but also I'm looking at genes. So
The samples Nick explained, he had this data where like you could like see like the PC1 was rotated compared to like the origin, right? Like that's like the example that he had. And like Nick highlighted how like you can project a sample back to PC1 and PC2. And so that's what we see here on the axis, right? PC1 and PC2 or the samples. For the genes, these arrows for the genes, the angle between the two tells you how correlated those two genes are. So like some of these genes over here have almost the same angle. So they're really highly correlated. A 90-degree angle between two genes would say like those genes are actually correlated. And you can kind of see that here with the genes for PC2. They're mostly uncorrelated with the genes for PC1 because either the genes for PC1 are either going up or down, and the ones for PC2 are like going mostly like flat. Now that's the angle. Then the length of the arrow gives you like a little bit of the idea, like the strength of the contribution of that gene. So in PC1, some of them are going down, some of them are going up. So they're completely like opposite angles. But like you could see that maybe PC1, this gene over here probably has the longest arrow. Whereas for PC2, I think is this guy over here. They're just like, since all of them are going in the same direction to the right, instead of having some of them to the right, some of them to the left, they're a bit overplotted, partially because these labels are pretty big. But there you can see that this gene is the one that's contributing most to that PC2. In terms of gene ontology, what you were asking, Luis, right? It's hard to do gene ontology on just like 10 or 5, whatever genes. Yeah.
Gotcha. I guess I was just wondering, like, this is neat, but I guess like what is there like a useful conclusion to draw from here? Just like we're just like, okay, like these are our 10 Gs for PC1. I guess it makes the PCs like a bit more understandable, but I guess I was just trying to think through like what's the application for like understanding our data better.
Yeah, like a biologist might know more about those 5 or 10 genes, right?
Okay.
I guess you could try to use the loadings as an input towards like a gene set enrichment analysis type of thing.
Yeah. Okay.
Instead of like, let's say like the t-statistics of a differential expression analysis, you could maybe use the loadings there. Does this help, Cynthia?
Yeah. Thank you.
So next question from Aaron. How is correlation developed in eigencore plot for categorical variables versus PCs? That's a great question. I hadn't actually thought about that. For obviously, if there's 2 categories, you can sort of make it numeric, but for more than 2 categories, sometimes study or I wonder if they might say sort of mention that in the details, but it's all lowercase. I'm not actually, that's a great question. Sometimes functions will really do a good job of like documenting exactly how each case it's handled and stuff, but I'm not sure. We'll get that. Maybe down in the examples for this. I don't think. Yeah. So to be honest, I'm not actually quite sure how those are handled. And offhand, it's not clear. Like I can't think of a way that would work. So I'm not sure. Sorry for not a great answer, but something to look into, I guess.
Yeah, that'll be fine. Thank you.
Yeah, so I think the rotation, actually, I forget which one we typically use downstream. So normally we have it samples by PCs. So that looks like what the rotation is, but I would have to actually check on that. So I guess the remainder of this, I haven't looked. There are other features in this package that I'll just like briefly mention that they exist. I haven't reviewed them too much. So one thing is like there's this find elbow point method that I want to talk about just very briefly. So you can give it the variance explained for each principal component, and it will try to find an elbow where you're explaining sort of the amount of variance that you're explaining drops off as you increase components past that point. So like in this example, they were saying like keep 8 components, and that looks good. So in this vignette, they say like actually there's not like a super great mathematically robust way to get an exact answer for this. So there's different methods that sort of yield different answers. They have, I think they also say that there's like this form method that is down here. So I guess something to keep in mind is that like there's different methods that have different sort of implementations, but deciding how many principal components to pick is not always like, it's not like there's a clear correct answer for that. But this package just provides a few different ways to do it, so that's useful. And there's a lot more, I think, that go more into sort of customizing these plots. I haven't checked this out too much, but just to mention that these are very highly customizable. You can even sort of try to draw circles around the observations to try to see how things separate by PC and so on. So yeah, let me make sure. Yeah, they have some other functions for circling data. Haven't checked too much into that. And yeah, I don't know. That's most of what I had for today. There's more to this package, but maybe that would be for another session to really dive into this. Because it seems like there's a lot to cover here. So yeah, I don't know if there's any questions. I guess we already covered a couple, but
Thank you, Nick.